Okay, so back to single cell. So what do we actually do to annotate single cell data? So again, we need some training data because any classifier needs examples to learn from. And these can be, atlases. There are a lot of large data sets that you can use now, but it can also be simply a data set that you have annotated at a certain point, and then you want to use it as training data for the next phase of your project or something. Some classifiers do rely on marker genes. I will talk about this more later. So they say, okay, I know that B cells have to be positive for this marker. I know that T cells have to be positive for CD3 marker, and so on. We'll talk about that more. And then basically that's it. You train your classifier. Once you get a new test data, an annotated data set, you run it through the classifier, and that will give you the labels. Some classifiers will also give you an ambiguous label, like an unknown, like I have never seen the cell before, so it might be something new or noisy, or I don't know what it is. So we'll talk also more about this. This is called more rejection option in Okay, there is a bit of a summary in the paper in terms of performance with these intra datasets, so these artificial experiments with the inter datasets. That's the more realistic scenario you would want. A bit on evaluation on the rejection and also the timing, how fast or easy is it to run certain classifiers. But just to summarize this, we think that simpler classifiers are better. So the more basic the classifier is, the more convenient is it. So we advocate for SVM in the exercises you're using scPredict, which I also think is a really great classifier as well. There is another preprint from Fabian Theis's lab that kind of says the same message, and they do more extensive cross dataset experiments. So definitely look at it. These are more realistic scenarios where you take one brain dataset, predict another one, but they come back to the same message that if you train a neural network or a linear classifier, then linear classifiers are doing better as well. Adding these prior knowledge or marker genes to the classifier doesn't really help in our experience with PBMC. We did not test this for other tissues, but at least for PBMC data, this did not help at all. Rejection is difficult. I think I've made my point here. We do have this snake-make pipeline. So if you develop a new classifier and you want to test it, you can easily run all the experiments we did on this classifier. Or if you say, okay, you haven't evaluated on, I don't know, kidney tissue, I am really interested, then just add your kidney data set and compare all the classifiers. So I think I have a bit of remaining time. So I want to tell you a little bit about things that are a bit more challenging. So as I said, I talked about the rejection, but there are also more challenges. So we rely a lot on this training data. So which training data should we use? This is a bit of a difficult problem. There are a lot of inconsistent labels. So you can say, I'm going to take all the PBMC data that exists in literature. But then you need to match the classes or the clusters between these data sets. So are you going to match them based on the name? So just take whatever people call CD4, merge them with another CD4 from another data set? Or is there a better way to do this? Sharing of data is an issue. So if you're really trying to collect a very large atlas, a lot of data set you will figure out are actually not shareable easily across different places. Perhaps you can check this paper again from Fabian Theis's lab. I think they did a really nice study on COVID data sets in the past year. But again, sharing this data, even within Europe, was particularly difficult. So these are like considerations that you have to take into account. Yeah, so just to summarize a little bit, I think in general, this cell identification is moving a bit from clustering manual annotation to more classification or learning. It's not there yet, I have to say, but I think that is where we would go with more data sets being generated, people wanting to compare data sets across different labs or conditions and so on. So you really want to trust your labels of data sets. We did this comprehensive benchmark. I hope it helps if you have a look at it to make a choice on what is best for my situation. If I have a lot of cells, do I have many populations? Do I have very overlapping populations? And we just put an idea on this continuous learning. If data keeps growing, how can we maintain that the reference that we're using can also grow with the data as well? So this is just a way to do this. I'm curious to see how this will develop. Yeah, these are the bunch of people that I work with. So all the classification work I showed is the work of Lika and Tamim. And yeah, if you have any questions, I'm happy to answer.